Many areas of research are classified as omics, involving analysis of proteins, RNA, nucleic and non-nucleic DNA, including methylated DNA, metabolites, lipids, or modified histone proteins and chromosomes. In proteomics, there are over 1 million protein isoforms, many of which are key components of cell function. Understanding how proteins function help us better understand human health and disease. Our approach is to monitor proteins as disease biomarkers in blood samples. Scientists can measure multiple proteins in blood and serum samples to develop diagnostic tests aimed at identifying different diseases. Proteins are measured using blood sample preparation workflows, combined with proteomic approaches using mass spectrometry to measure hundreds of potential biomarkers simultaneously. This can be done with high specificity by using a few microliters of blood or serum. However, a challenge facing omic researchers is the cost and logistics of cold shipping a sample to the lab. When collecting wet samples in different countries for collaborative studies, the wet samples must go through customs, which leads to delays. It is easier to use dry blood serum microsamples that don't require cold shipping. Microsampling devices with absorbent tips can be applied to the surface of wet blood or serum samples in a lab to collect a precise volume. These microsamples dry on the devices, which then can be mailed via regular post to a collaborating lab. Dried samples remain stable at ambient temperatures for a few weeks, so mailing and storing them is easier. Microsampling devices make it easier to conduct omic studies using samples from a broader range of people, including those who live in distant locations or underserved populations.